I'm Valentina Greco, arrived in 1998 for an interview, and there was a number of Sicilian, and specifically Palermitan, that had arrived here at the EMBL. So they opened me to the existence of this amazing place and the possibility of doing science outside my home. And so thanks to them, the bridges that they had already created, the presence they had created at the EMBL, for which they were perceived as productive, integral part of this place, I could go through a selection, being interviewed, making it to the final, and then joining September 1998.
So how was your impression of the EMBL when you came?
Yeah, so I liked research before coming. I was doing cell culture and trying to understand division and cell cycle. But I remember during the interview, I've spoken also in the past about, for instance, Michael Way showing me this listeria infecting the cells, and in general the incredible microscopy that was present in cell biology, the model organism in developmental biology. It felt like a playground where I could have fun forever.
Mm-hmm. Okay, so what did you get involved in when you joined the PhD program?
Yeah, during the interviews I asked Suzanne Eaton if she would consider me, and as you remember, it was a matching system, and so Suzanne endorsed my application, and so I ended up being very lucky to do a PhD with this pioneer in developmental biology, cell biology, in many different fields. And so with her, she helped me explore how organs maintain their size, reach their size and maintain their size by focusing on epithelial cells. And so I was very fortunate to have a project that was at the interface between cell and developmental biology and looking at how epithelial cells communicate at distance through the engagement of morphogen and the dispersal through membrane vesicles.
I remember we were a class, and so there was always some friends around, but do you think that this organization of the PhD and the spirit of the EMBL helped you during the PhD, especially the colleagues, the other young guys?
Oh, absolutely, spot on. I think the human environment was really fundamentally inspiring and also supportive. So like you said, we were a class that was all immigrants, essentially, and so I remember we were speaking to each other in English because there were some few that really knew English and some others that had adopted as second or third language. And it was, even during the interview, I felt that with most of the people were extremely supportive to each other. You could imagine right there, some sense of competition could make us fall apart. But I formed a friendship already during the interview and then throughout the class for all the trips that we were doing together.
So, but then, of course, you know, there is life after the EMBL. You move to another position and other projects. So what did you do after the EMBL?
Yeah, so after this imprinting of cell developmental biology, I was fascinated by trying to understand tissue biology and multiple more cell types. So I went and took a training on skin and hair follicle. And that was a way to expand, but also change a bit the field. I think a change in the scientific discoveries was important to me because it could challenge me in having to adapt my thinking, evolve my thinking, and also expand the knowledge that then I could combine later on. And so that knowledge about skin, for instance, and hair follicle, combined with the microscopy of my PhD, allowed me to seek collaboration at the beginning of my lab when we launched it in August 1st, 2009, to now start to see, can we try high-risk, high-rewards type of projects? And so thanks to an intravital imaging facility at Yale, and Dave Gonzalez and Aberman and many others, we were capable then of creating a new contribution in the field. Mm-hmm. And I think this is stemming from the fact that the understanding, having multiple expertise and combining them when they have not been combined yet, is an edge for making contributions later on.
Which I think is another lesson learned from the EMBL, with this mixture of different discipline and the technical part supporting the science, and the science helping developing the techniques.
Couldn't agree more. EMBL was fostering interdisciplinary approaches, and Suzanne Eaton in particular was always teaching me how drawing from different fields, whether it's physics, philosophy, history, can always allow you to see problems with new lenses. Mm-hmm. And that creates the excitement that then move forward the field.
Mm-hmm. So what are you passionate about now in your science, now that you have a beautiful and successful lab?
You know, the way we look together with the members in my lab and I, the cells that we study at, are with principle of community. I really draw from understanding how people interact with one another and people behavior as an expression of the system they navigate, the same way we look at the cells. What is their behavior telling us about the principle by which the tissue have to function, by which the tissue have to be organized with their architecture and shape. And so we integrate back and forth between life and what we see through the microscope. And what we like to understand is how can we have a more hypothesis than the one we are always constraining ourselves within. And by that, I mean the society gives us a language and a narration that is very narrow, sometimes winner and loser, sometimes ranking order of groups. And I think that constrains the way that we can actually make discoveries. And so we continuously scrutinize ourselves for what are we being narrow about, what we should be expanding, and applying it to the process of doing research.
Yeah, I mean, it's interesting that you can see the same principle on very different realities, but I think, you know, this happens a lot in life and in biology. You have a sort of repurposing and similar, I think, you know, needs, in a sense. The environment imposes similar needs, and so you come up with similar logical solutions. I know you're also very passionate about not only STEM and science in general, but also on the issue of gender in science and minorities and women in science. So can you tell us a little bit where this originates from and, you know, how you're going about these issues?
Absolutely. So not surprisingly originates also from lived experiences, having watched incredible pioneers like Suzanne, Eaton, and many others not being recognized the way I think they should have been recognized. And often I find myself wondering, if I were a male, would that have happened to me? Or even comparing directly notes with my husband, Antonio Giraldes. So many times we've been in conference together, I would ask a question and the person would turn to him to answer. Or we would win the same awards and we would be interviewed for the same amount of time, and his quotes were much more prominent and mine were cut. So it's constant and pervasive. And it is really exhausting because some people sometimes react in saying either that you're playing the victim or it's all in your head. And so it's a perpetual cycle of feeling clear about what's happening. But that clarity often is understood and accepted by people in my own membership, or membership that are even treated more poorly than mine is. Because there is an intersectionality within our groups that is not only gender, is power construct of race, power construct of sexual orientation, of abilities, you know, religion. And so I dream of a world that doesn't exist yet, but where I think we can ally across groups, because we really can be so much more powerful if power was equitably distributed. But it takes for people that sit on privilege to have to give up a bit of that privilege, and that might be difficult.
Yeah, no, for sure. And one thing that I was reflecting on along these lines is that, you know, you talked about a system that was designed in a certain way and certainly doesn't accommodate this diversity. But the other feature of the system is there's many, you know, elements to that. And, you know, that these little inequalities are not really only summing up, but they are sort of self-amplifying. So I guess one aspect is, you know, really being able to recognize all these little pieces and act on multiple, because there's not a single intervention that will cut it. So maybe you can tell us a little couple of ways in which your lab is trying to support the diversity and minorities.
Absolutely. I really love your point of self-amplification, because I think it's fundamentally important, and we think each step is benign, but it can really lead to incredible internalization of inferiority, even though it's tall inferiority. For instance, even in this amazing conference, if you hear talks, more often than not, male will continue to amplify almost all names of males. Whereas if you hear talks from females, you will see amplifying the names of females and males. And it's not to say that we are not all slave of sexism, because if the power structure is such that it is male-based, we all orient to power. So this sample amplification is really dangerous. And so in the way we have been doing in my lab is one, continuous self-scrutiny and continuing courses for me. For instance, I should be acknowledging that I have internalized sexism and racism and many other structures all the time. So when I'm writing, for instance, an invitation to two students that want to join my lab, knowing that one is female, one is male, before sending the letter, I read it. And guess what? I also found that exactly like society has told me to be more differential towards a male and less towards a female, the imprinting was there. And so I could go on, look in the face, correct it. But if I, who lives on my own skin and sees it outside, don't make these pauses, I don't think I'll be able to correct myself and orient in the right direction. So it takes activism on every single step we make. The way that we mention the work from a female versus a male, person that is white versus non-white, all that we have internalized about stereotyping of all these groups, for us to really be able to capture the creativity that comes from this different socialization. Because each socialization has an incredible power. For instance, being socialized as a woman, we come up with the ideas of community and interaction between mutation in the skin that is collaborative. That made us won a Pioneer Award. So it's not that there is no business for all our socialization, there is, but there is not enough space for all of us to bring what our socialization has brought, because there is too much benefit to keep the system as it is right now.
Yeah, no, sure. I mean, it will be a long way. But what I wanted to ask you is, because now you've been living for quite long in the States, and the States have, maybe to some, extent, a larger minority community, an older minority community than many countries in Europe. So, did you develop this sensibility by living in the States, and do you see differences being a European woman between how these issues are dealt with in Europe versus in the States, and also in science, of course?
Thank you for that question. I find that there's two important parts I would like to respond to. First of all, if I can reflect on the language we use for minority, if we think about and we consider, for instance, the white male as actually the dominant group, all the rest is a majority. But see, we have been so pervasive in our language that we call all this 90% minority. And that already shows you how we want to control it, oppress it, and suppress it. So if we speak about can we give space to all of them, and Valentina, have you increased your awareness because in the social context you are in is more rich than here? I think I came there because of many roles I play, for instance, Vice Chair of Diversity, for which I took an anti-racism course, and also movement of DI office that had been put in place in university, made me realize my internalized racism, for which actually back home is not that different. Like when I think about the mixing and the lack of mixing, is structurally imposed. And I think maybe might not always take a place as many different culture or nationality, but it can take place as classism, because it's the roots of capitalism, right?
Yeah,
so we
absolutely.
always manage to structurally exclude the majority in many different dimensions. And the reason that we turn to it is when we realize that one, there is a malfunctional system, when we are victim ourselves, when we straddle privilege on both one side of privilege as well as the other. And so my reckoning is my own difficulty in going forward, in which I realize not only I want to do something for my own group, but also for all these other groups that collectively are the majority of the population.
Yeah, I mean, again, along these issues, but coming a little bit more to science, I think, there is, and you are a good example of that, also inequalities in access to possibilities, right? I mean, you are an example of coming from maybe a region that doesn't have a very well-developed science, but has a lot of talent in terms of students. And so how do you, I mean, are you conscious of that? I guess you are, but how do you go about selecting talent that might arise, far from Boston or far from Heidelberg and everywhere?
Yeah, I think for all of us, when we do hiring and we start to interact with people, I think having a moment of recognition, what is the ranking order we have absorbed from society, even the language the single-handed experiment versus the narrative of actually being a collective. How people start to talk to you can be great insights of how they think about team approach. And by team approach is not to mean that the person is not actually capable on their own, but how they understand community. Second is their passion and enthusiasm. Third is a normalization process to understand what they've accomplished based on the type of resources that they come from. And so if we only select for top journal and first author papers and amount of time to reach those journals, we are essentially missing incredible perspective. And so having that normalization, I think, is fundamentally important.
Yeah, so you're saying we have to find ways to take all that into account when we select. And I mean, in this, I think, you know, Suzanne is a great example of somebody that was able to see through this thing and recognize it's a story that you told many times, and recognize talent irrespective of where it was coming from. Okay. Great. So maybe we'll transition into a couple of other questions about the EMBL and the community. I mean, the alumni community is a huge community. Is it something you're still tapping nowadays after many years that you have left the EMBL?
Absolutely. I have dear friends that have both studied in the United States and then come back here. For instance, neurobiologist Chaleroglu at Duke, we are still in touch to actually this week. Even though the disciplines are not exactly the same, I really admire her work and continue to follow her closely. Cell biologist Michel Bagnat, another incredible scientist who was actually my neighbor and who spent time teaching me about genetics while I was being on board at the EMBL. Thomas, Shell, Celia, Antonio, Tobias, Walter, there are so many. And Thomas, of course, you and I have been also in the same class, and also later on when you joined.
No, I mean, I absolutely agree. I mean, it's essentially a lifelong web of relationships.
Definitely.
Okay, so that's great. I'm gonna ask you one last question that is related to the 50th anniversary of the EMBL. So the question is, what EMBL means to you?
Yeah. It has opened myself to the love that I cherish for science and research. I think until EMBL, I liked biology, I liked research, but I really fell in love, and it became like being in a candy store, the possibility of all the questions one could ask when the creativity was used to think about solution having resources, rather than having to use creativity to find resources.